Viruses are everywhere, in the air we breathe, the water we drink, and even inside our bodies. They are incredibly small, much smaller than bacteria, and can only be seen with powerful microscopes. Unlike bacteria, which can reproduce on their own, viruses need a host cell to survive and replicate. They are essentially tiny packages of genetic material, either DNA or RNA, encased in a protein coat. When a virus infects a cell, it takes over the cell's machinery and forces it to produce more viruses. This process is called the viral replication cycle. Understanding this cycle is crucial for developing antiviral therapies and vaccines. Imagine a microscopic hijacking where the virus is the cunning thief and the cell is the unsuspecting factory. The virus breaks in, seizes control, and uses the factory's resources to produce thousands of copies of itself. The study of virology, the branch of science that focuses on these intriguing entities, has revealed a great deal about the stealthy tactics viruses employ. These insights are not only fascinating but also critical in our fight against viral diseases. By understanding how viruses replicate, we can develop strategies to interrupt their life cycle and prevent them from causing harm. This essay will take you on a journey into the microscopic world of viruses, exploring the fascinating steps involved in their replication cycle. Viruses are highly specific in their choice of hosts and the cells they infect. This specificity is determined by the interaction between proteins on the surface of the virus and receptors on the surface of the host cell. Think of it as a lock and key mechanism. The virus has a specific key that can only unlock a specific lock on the surface of a cell. For example, the influenza virus, which causes the flu, primarily infects cells in the respiratory tract because it has proteins that bind to specific receptors found on the surface of these cells. Similarly, the human immunodeficiency virus, HIV, which causes AIDS, specifically targets cells of the immune system because of its affinity for a receptor called CD4, found on the surface of these cells. This specificity is why you can't catch the flu from a plant or HIV from a mosquito bite. The virus simply cannot find the right lock to attach to on the cells of these organisms. Understanding this lock and key mechanism is crucial for developing antiviral drugs that can block viral attachment and prevent infection. In the next section, we'll delve into the various ways viruses gain entry into host cells, bypassing the cell's defenses and initiating the next stage of their replication cycle. Once a virus has identified and attached to its target cell, it needs to find a way to get inside. This is a critical step in the viral life cycle, as without entry, the virus cannot replicate or cause infection. Viruses have evolved a variety of clever strategies to breach the cell membrane and gain entry. These strategies are the result of millions of years of evolution, fine-tuning the virus's ability to infect host cells. These strategies can be broadly categorized into three main mechanisms: direct penetration, endocytosis, and fusion. Each method has its own unique way of overcoming the cell's defenses. Direct penetration is like a brute force attack. It involves the virus forcing its way into the cell. Some viruses, such as the poliovirus, have specialized proteins that can create a pore or channel through the cell membrane, allowing their genetic material to be injected directly into the cytoplasm of the cell. This method is highly efficient and ensures the virus's genetic material reaches its target quickly. This is a quick and efficient way to deliver their payload, minimizing the time the virus spends outside the cell and vulnerable to the host's immune defenses. Endocytosis, on the other hand, is a more subtle approach. It involves the virus mimicking something the cell would naturally take in. Viruses like the influenza virus take advantage of the cell's natural process of engulfing external particles. This process is usually reserved for nutrients and other essential molecules. The virus tricks the cell into thinking it is something harmless, like a nutrient molecule, and the cell engulfs the virus in a small pocket called a vesicle. This vesicle then transports the virus into the cell. Once inside the cell, the virus escapes from the vesicle and releases its genetic material. This allows the virus to begin the process of hijacking the cell's machinery. Fusion is like a merging of two entities. It involves the virus's membrane merging with the cell's membrane. Enveloped viruses, such as HIV, have a lipid membrane surrounding their protein coat. This lipid membrane is similar to the cell's own membrane. This membrane can fuse with the cell membrane, allowing the virus to enter the cell. This method is particularly effective for viruses with a lipid envelope. Think of it like two soap bubbles merging into one. The contents of the virus are seamlessly delivered into the cell. Each of these entry mechanisms highlights the remarkable adaptability of viruses in overcoming the challenges of infecting host cells. They have evolved to exploit the cell's own processes to their advantage. In the following section, we will explore how viruses, once inside the cell, hijack the cellular machinery to replicate and produce more viruses. This is where the true battle between the virus and the host cell begins. Section 4. Hijacking the cellular factory, reprogramming for viral production. Once inside the host cell, the virus's primary goal is to replicate its genetic material and produce more viruses. This is a complex and highly coordinated process that involves several steps. To achieve this, the virus must take control of the cell's machinery, converting it from a factory for producing cellular components into a factory for producing viral components. This transformation is both fascinating and alarming. The virus's genetic material, whether DNA or RNA, carries the instructions for building new viruses. These instructions are like a blueprint for constructing new viral particles. The virus releases its genetic material into the cell, and this material enters the cell's nucleus, the control center of the cell. This is where the real takeover begins. Here, the viral genetic material interacts with the cell's own DNA and takes over the cell's protein synthesis machinery. This interaction is crucial for the virus to hijack the cell's functions. The cell's ribosomes, which normally follow instructions from the cell's DNA to make proteins, are now tricked into following instructions from the viral genetic material. This deception is key to the virus's success. The cell begins to produce viral proteins instead of its own proteins. This shift in production is a clear sign of the cell's hijacking. These viral proteins are the building blocks for new viruses. They are essential for the assembly of new viral particles. Imagine a factory worker who normally assembles cars suddenly being forced to assemble bicycles because the factory has been taken over. This analogy helps illustrate the drastic change in the cell's function. The worker is still skilled at assembling, but the instructions have changed, and the output is now different. This change in instructions is what the virus does to the cell. This hijacking of the cell's machinery is a crucial step in the viral replication cycle. Without it, the virus cannot reproduce. In the next section, we'll see how these newly produced viral proteins are assembled into new virus particles. This final step completes the viral replication process and prepares the virus to infect new cells. Section 5. Building new viruses, assembling the pieces. In this section, we delve into the intricate process of how viruses assemble themselves within a host cell. With the cell's machinery now under its control, the virus directs the production of all the components needed to build new viruses. This hijacking of the cell's resources is a masterstroke of viral strategy. These components include copies of the viral genome, which carry the genetic blueprint for the virus, and the protein subunits that make up the virus's outer shell, called the capsid. These proteins are crucial for the virus's structure and function. Think of it like an assembly line, where each component is carefully crafted and then put together to form a complete virus particle. This process is both precise and efficient. The viral genome, containing the instructions for making new viruses, is replicated many times over. This ensures that there are enough copies to be packaged into new virus particles. These copies of the genome will be packaged into new viral particles ready to infect other cells. Meanwhile, the protein subunits that form the capsid are produced in large quantities. These proteins are synthesized by the host cell's ribosomes, which have been commandeered by the virus. These subunits spontaneously assemble themselves around the newly replicated viral genomes, forming new viral capsids. This self-assembly is a remarkable feature of viral biology. The capsid acts as a protective shell, safeguarding the viral genome from damage and ensuring its stability. Some viruses, known as enveloped viruses, have an additional outer layer called an envelope. This envelope is derived from the host cell's membrane. And is acquired by the virus as it buds off from the host cell. The process of budding allows the virus to exit the cell without immediately destroying it. The envelope helps the virus to evade the host's immune system. By mimicking the host cell's membrane, the virus can go undetected for longer periods. This assembly process is remarkably efficient, and soon, the host cell is filled with thousands of new virus particles, ready to be released into the surrounding environment to infect other cells. The sheer number of new viruses produced can overwhelm the host's defenses. In the next section, we'll explore the different ways viruses escape from the host cell and spread to new hosts. This is a critical step in the viral life cycle, ensuring the continuation of the infection process. Section 6. Escape and spread. The virus breaks free. With the host cell now packed with progeny viruses, the final step in the replication cycle is release, where the newly assembled viruses escape from the infected cell to infect other cells. There are two main mechanisms of viral release: cell lysis and budding. Cell lysis is a dramatic process that results in the death of the host cell. The infected cell, now filled with viruses, swells and bursts open, releasing the new viruses into the surrounding environment. This process is often compared to a balloon bursting after being overinflated. Budding, on the other hand, is a more gentle process that allows the host cell to remain alive, at least for a while. Enveloped viruses such as influenza and HIV acquire their envelope by budding off from the host cell membrane. Imagine a small balloon being pinched off from a larger balloon. As the virus buds off, it takes a portion of the host cell membrane with it, forming its envelope. This process can continue for some time, with the host cell releasing viruses like a factory churning out products. The released viruses are now free to infect other cells, repeating the replication cycle and spreading the infection. In the next section, we'll explore the diversity of viral strategies and how different viruses employ unique tactics throughout their replication cycle.